lecturer, Dr. Rashidin, and to all my fellow colleagues. So I'm Oliver, this is Aida Karina, and this is Nick Lawrence. Today we are going to present our research proposal, which is about micropropagation of Ananas commusus L species using periodic immersion bioreactor. So this is the abstract. In this research, we are going to genetically modify the generic DNA of pineapple to be resistant towards non-selective herbicide. In the second part, we would like to implement the micropropagation to commercial the pineapple due to the rapid rate of production. And then the purpose of this production type is that we want to produce this type of fruit which has the herbicide tolerant trait and will not get easily infected with herbicide usage. With the discovery of the bar gene from S. Hygroscopius bacteria, herbicide tolerant among these pineapple will be achieved and even possible. Through the mediated transfer, we can introduce the bar gene into the pineapple, and then the agrobacterium will carry the gene, infected the cells of the another pineapple, and then we can produce large quantity of pineapples with the good traits. By the invention of periodic immersion bioreactor system, which will enable the rapid production of transgenic pineapple plant, is our second objective towards the success of this project. For the introduction, so scientific research and management of environmental productivity is the key towards the sustainable development of the agricultural sector, which we want to have the high yield based on the quantity and also the quality of the crops. Their manipulations and variations are to be taken into consideration. So we want to have the vast management of budget, so we propose to use micropropagation method. It is a multiplication system and use some techniques such as induction of auxiliary and adventitious shoots, the culture of isolated resistance, and also plant regeneration, extended bacteria, and all. And also, what we choose is Ananas commusus species, which is a species of pineapple, to keep it sustainable and highly demanded to meet the market demand. Our objectives are to modify the Ananas commercial species to the desired traits of herbicide tolerant for better quality of crop yield, and to implement the effective usage of micropropagation to produce Ananas commercials in large scale commercialization.
So for the literature review, commonly for the multiplication rate of pineapple is 11 to 17 plants in a period of 5 months, which is very little. So what we are going to emphasize is the mass propagation of the pineapple within a very short period of time with high quality and high quantity. So in this study, we are proposing that the production of a new variant of pineapple that are herbicide tolerant, as well as lowering the cost of production. So from a study of Sri Pauraya, whereby they use the initiative of biologistic approach using microprojectile, which utilizes the bar transgene, which changes over herbicide into non-lethal acetylated form, a study from the Hillary. There are also reports on using agrobacterium infections as a method of gene delivery. There is this one research done by Graham, whereby a black heart resistant transgenic plants is produced via both microprojectile and also agrobacterium mutations. So that was for what we actually did for genetic manipulation. micropropagation. So to reduce the cost of micropropagation, there are actually lots of research actually being done by using natural light conditions of a net house, tubular skylights, a substitute for inefficient lights, mechanized self-filtering, and also usage of periodic emission bioreactors. So, B and Diverge studied that the utilization of hormones also likewise to impact the usage, the cost of production, whereby in rapid production, whereby number of hormones actually, the amount of hormones actually manipulated their production. Higher number of hormones actually gives a lower quality and actually decreases the size of the pineapple itself. So a lower amount of hormones actually is needed for production of pineapples to make them high quality. For countries like tropical regions such as southern Vietnam, Thailand, and Malaysia and Indonesia, they actually has a good rate of natural sunlight throughout the year, where it actually will have a lower production cost. So we propose usage of agrobacterium delivery and propagate them using PIB, which are partial immersion bioreactors. So it can actually reduce the cost of production through 35% compared to the traditional method based on study of feasibility and Dutson. So through this, we can help actually farmers by supplying better variants, which are easy to take care and have a higher production.
Okay, thank you. Next, I'll be talking about the methods and materials that are required in our experiment. So, first of all, is the first methods are the isolation of Galapagos-resistant genes or the bar genes which are originate from S. hygroscopicus species, and then introduction of the genes into the target plant, which is the pineapple plant cell, and then this is of course via the agrobacterium mediated transfer. And then is the next mass production of transgenic plant through the micropropagation. Okay, so first of all is the competent cell preparation. So these are the steps required. So basically, we are just going to take a stock of agrobacterium and dilute them. After that, we're going to incubate. First of all, we're going to spread the stock agrobacterium on LB broth. And then we're going to incubate at a room temperature and grow to OD of 0.5 to 1. And then we're going to chill the stock on ice for 15 to 30 minutes before we centrifuge, and the pellet we're going to resuspend in a 90 microliter ice cold TSS buffer. Okay, so next is the isolation of the TI plasmid from the agrobacterium television. Okay, so next we're going to talk about resistant enzyme digestion. So at this step, both the TI plasmid and also the target genes, which are the bar gene, are going to digest using the same resistant enzyme, so that they have the same restriction cut. Okay, next is the ligation of the bar gene into the TI plasmid. After we digest them using the restriction enzyme, we are going to ligase them as the following. So the following reaction will be set up as 5 microliters of 2-time rapid ligation buffer, TI plasmid, bar gene, and also T4 ligase. Okay, next is the transformation. Okay, after we manage to successfully ligase both of the TI plasmid and also the target gene, the bar gene, we're going to transform them into the target cell, target host, which means that we are going to put the recombinant TI plasmid into the agrobacterium. Okay, after we manage to insert the recombinant TI plasmid into the agrobacterium, and then we're going to introduce the agrobacterium into the host, which is the plant cell. This is we allow them to be infected. Okay, after we manage to insert the recombinant agrobacterium into the host, into the plant cell, we are going to proceed with the micropropagation, which means we are going to do micropropagation using the PIB, periodic immersion bioreactor, to mass produce the transgenic plant.
So we are going to expect to have the desirable traits in the crop yield of the pineapple, and through the micro cultivation, we hope to produce vast amounts of pineapple to meet the market demand. So I hope that our objectives will be achieved. So that's all for us. This is our schedule. Thank you.